opportunity to greet them, and I feel honored for that. Welcome to all distinguished persons and researchers from different institutions, along with our students, as well as I would like to address the professors, lecturers from different universities, those who have attended here. We express our sincere gratitude to our Honorable Director Sir, Dr. Pradeep Ghosh, the Chief Patron of this workshop. He didn't attend here due to his busy schedule, but we always feel his presence in everywhere. His encouragement and assistance and support always haunt us to do new things. Such kind of workshop or seminar are going on from the day beginning since the year 2017. We are also deeply indebted to our Principal Sir, Dr. Sudipto Chakraborty, and also Vice Principal Sir, Dr. Kuntal Ghosh, that is patron and co-patron of this workshop, for their suggestion and advices time to time. Sir, really we are grateful to you. Greeting to all resource persons for this workshop. Sir, we are delighted and enlightened by your presence. We are very pleased that our respected Vice Principal Sir, along with us, Dr. Kuntal Ghosh, co-patron of these workshops. This 2 days workshop will cover regarding the collection, preservation and identification of techniques of algae, fungi, bryophytes, orchids, focusing on the species identification, ecological importance and preservation. Though these organisms are different from each other, but they played the important role in ecology, particularly in ecosystems, maintaining the ecological balance and biodiversity. Generally, we overlooked, but their roles in a biodiversity or ecosystems is very important. Our veteran expert scientist will delve to find out the nuances among the species and will help to identify them and give us the information how to preserve and their role in a biodiversity. That is, algae and fungi bryophytes, all are the essential parts of ecosystems. That carbon sequestration or oxygen generation, the major role played by algae, and contributing near about 40% oxygen in the atmospheric environmental. At the same time, we address the algae as a planet's lungs. In case of fungi, this also addressed as an unsung hero. In terms of nutrient recycling, soil health, this played a vital role. Sir, really we will be grateful getting enough information from your end, and we are eagerly waiting to learn to this information from your end. At the same time, I also not measure to mention that is the orchid, that we generally laymen address them as a ornamental plants, but it's a sign of healthy ecosystem and support ecological balances. So, speaker particularly encourages all the participants, those who are attending here, they should involved directly, and they also share their knowledge how to preserve them and their importance in the ecosystem of these organisms. So, we are eagerly waiting for the participant to ask some question related to this workshop. This workshop aims to raise public awareness, conduct research, develop conservation solution for these vulnerable organisms. Vulnerable in that sense due to overexploitation or shelter loss, pollution, overpopulation. These teeny creatures are deteriorating day by day. This is also alarming situation if we do not give attention for their conservation or preservation. So these workshops will give more information about this. How we will conduct, that is by our discussions, that information will percolate to the layman's, or we can share this information in other persons, and discussion and partnership formation, that will also a good window that we can percolate this information. Everyone, regardless of their profession, whether is a student, researcher, or any post holds, they play an important role for the cooperation and creativity and moral dedication, particularly moral dedication, how to preserve or conserve them. This is the importance of these workshops. We will learn later on, and the next part I would like to request our student Srijoni to continue it.
Thank you, sir. Gratitude can transform common days into thanksgiving, turn routine jobs into joy, and changes ordinary opportunities into blessings. Hence, it is a ritual to felicitate our respected guest, and I would like to invite our Honorable Principal Sir and Vice Principal Sir and today's dignified resource person to the dais. Please. For this, I would like to call my juniors, Mahazabin Kothakoli from UG 4th SEM, Suchana, Suranjana, Smiti and Tanya from UG 2nd SEM to felicitate our guest. Sir, please. Thank you, sir. Thank you, juniors. I feel honored to invite our Honorable Principal Sir, Vice Principal Sir to participate in the illumination of lamp that symbolizes success. And also, all dignified guests are being invited to participate in this ceremonial occasion. Sir, please.
Thanks, Srijani. Next, I would like to request our Honorable Vice Principal, sir. Please, sir, say something about our esteemed college.
Honorable Director Sir, Midnapore City College, though he is not present here, but he expressed his best wishes for successful completion of this workshop. In the dais, respected Principal Sir, eminent scientists from Botanical Survey of India, my colleagues, dear students and delegates. A very good morning to all of you. On behalf of the Midnapore City College, I would like to cordially welcome you to this 2 days workshop on hands-on training on collection, preservation and identification of orchid, algae, fungi and biophytes, jointly organized by Department of Biological Sciences, Midnapore City College and EIACP PCRP on Biodiversity Flora, Botanical Survey of India. We are very much fortunate to have the eminent speaker from the Botanical Survey of India. I welcome Dr. R.K. Gupta, Dr. Kanad Das, Dr. Sudesh Nadatto, Dr. J.S. Jalal, Dr. Soshi Kumar and Ms. Midnapore. I welcome all of the participants to the symposium. Now I am going to give you a brief introduction of our college. Midnapore City College is a self-financing general degree college in West Bengal, recognized by the UGC, DSIR, AICT, Higher Education Department, Government of West Bengal, Director of the Medical Education, Government of West Bengal, and affiliated to the Vidyasagar University and the West Bengal University of the Health Sciences. Currently, 20 UG courses and 22 PG courses are being taught in this college. A Biodiversity and the Environmental Studies Research Center is also running in this college under the Vidyasagar University. The college received 10 extramural research grants from different funding agencies like ICMR, ACRB, DBT, Government of India. The faculty members and the research scholars published 125 research articles and book chapters in various national and the international journals of the publishers. The college signed MOU with the Raj University of the Medical Center, Chicago, USA, and Jeonbuk National University, South Korea for promotion of the research activities. The workshop aims to increase the awareness of the importance of the orchid, algae, fungi and biophytes in the ecosystem and biodiversity among the academics, ecologists and environmentalists and students. Since we are dealing with the high unhead of the environmental issues like shifting climate patterns and dwindling habitats, we must investigate and protect the species. This workshop will educate the researchers and students about the preservation and identification of the orchids, algae, fungi and also the biophytes. So we are very much thankful to the BSI Government of India for accepting our invitation. Without their generous support, it would not been possible to organize. I am very much thankful to Dr. Pradeep Ghosh, Founder Director of Midnapore City College for continuous support and advices which have greatly helped towards the successful organization of this workshop. My thanks to Dr. Supriya Chakraborty, Principal, Midnapore City College for his active cooperation. I thank all of the invited speakers and the delegates for their enthusiastic participation in this workshop. And a special thanks to Dr. Soumitra Pal and his team, including Dr. Anulya Manna, Dr. Tapun Das, and all of the faculty members for organizing this workshop. I also acknowledge the unwavering support received from all of the faculty and the staff members of the Midnapore City College. Hope you will enjoy this scientific session. Thank you.
Thank you, sir. Now, let's inaugurate the program with a melodious song. And for this, I would like to call Suchana, Suranjana and Tania.
Thank you, my dear student. Yes, Almighty, O God, you have sent us angel in the name of scientist. Really, we are eager to learn something new things related to this amazing creature in this earth, and we are excited getting with you. Let this session serve as inspiration for more initiative and strengthen collaboration. If we work together, we can change things. Let's commit to protect the biodiversity of our world for coming generation. And I appreciate you and looking forward to our time together and provide conservation and project that will result from it. And last not the least, I would like to appeal to the concerned authority of BSI. Please fix a day in your academic calendar for Midnapore City College to conduct such kind of workshop, seminar. Really, we are eagerly waiting for your consent so that we can allow maximum student, because our roll strength near about 600, above 6,000. So you can convey this message, information through large number of students. It is my appeal. I request you, please fix a day every year in your academic calendar. Thank you, sir. Today's session, we split it into two phases. The first session will cover Dr. Konad Das. Specifically, he will discuss about the identification of macrofungi. Prior to discussion, it is needed, and I feel honored to introduce himself in front of our student, the brief CV of Dr. Konad Das. Completed his PhD degree from Botanical Survey of India and HNV, Garhwal University, and postdoctoral research from the Agarkar Research Institute, Pune, India. Dr. Das joined BSI in 2008 as a scientist C. Currently, he is working as an additional director grade scientist F in the Central National Herbarium of BSI. Since his beginning of his research career, his work had been focused on microfungal exploration, collection, and characterization, identification, discovery, and documentation of wild mushrooms, particularly the Basidiomycota groups. With the help of conventional morphotaxonomy and single to multigene molecular phylogeny, while executing his research till date, he has discovered 2 novel genera and co-author of 150 new species of wild mushroom from India, which are being published by him in 155 original research articles, 7 books and 6 books chapter. 5 research scholars have already accomplished their PhD under his supervision. In 2015, he was awarded with a Bharat Jyoti by India International Friendship Society, and in 2020, he was conferred with a Professor K.S. Thend Medal by the Association of the Plant Taxonomy and Fellow in 2023 by Mycology Society of India for his contribution for fungal taxonomy. Thank you, sir. Please continue your session.
Respected principal and vice principal of this esteemed college, teachers, faculties, my fellow colleagues and senior scientists from Botanical Survey of India, and students from different corners. Ladies and gentlemen, good afternoon to all of you. Just tell me one thing, am I audible? If not, then please take these seats. There are several seats, so you can take this, right? Let me start with a few lines of Kavi Guru Rabindranath Tagore. বিপুলায় পৃথিবীর কতটুকু জানি, দেশে দেশে কত না নগর রাজধানী, কত নদী গিরি সিন্ধু মরু, কত না অপরিচিত জীব, অপরিচিত তরু রয়ে গেল অগোচরে. Students, you know, we have enormous and life and limitless biodiversity. We are trying hard to know only a little bit. And the unfortunate thing is that still we do not know what we have. In this direction, Botanical Survey of India, the premier institute of Government of India, and its ENVIS team, they are continuously trying hard to unveil or uncover the hidden treasure of biodiversity, especially on plants and fungi. Our scientists on different fields, they are working day and night to know what we have. Today, we are here only to, you know, in one day we can't make you a taxonomist, we can't make you a scientist, but at least we can give you the essence, the flavor, we can show you some directions, because you are the future scientist. You have to go ahead and you have to discover so many unknown things. I am Konad. I am working on wild mushrooms since last 25 years. And my today's talk is diversity, systematics, and identification of wild mushrooms. Before I go deep, for the convenience of the audience, you know what is taxonomy. But still I define, you know that it's a natural instinct, or you can say basic instinct. That is, we desire to sort out things into groups. Remember those days when we used to collect coins, we used to collect stamps, and we used to classify them based on their countries or based on the years. So this is something, this is our instinct, that we desire to sort out things into groups. So what is classification? Grouping things together on the basis of features they have in common. Hope you agree. And the taxonomy is nothing but the science of classification, which have two functional areas. One is nomenclature, that is naming of organisms, and the systematics, placing of organisms into groups. you know, since long back and for a long time, fungi was classified, grouped along with plants. And that is why still in our curricula, we have fungi with botany. It was only 1969 when Dr. R.H. Whitaker, the renowned plant ecologist who proposed the 5-kingdom classification of living organisms, and he, for the first time, he placed fungi as a kingdom. So fungi, the mycologists, for the first time, fungi got its proper place. Today we know that they are placed as a separate kingdom. They are no more plants. And you know that his monumental publication was published in the renowned journal Science in the year 1969. So coming to the kingdom fungi, you know the kingdom fungi is a monophyletic group. Do you know what is monophyletic group? Monophyletic group means that all modern fungi can be traced back, or they are originated from a single ancestral organism, right? So kingdom fungi is a monophyletic group that diverged from a common ancestor with the animals about 800 to 900 million years ago. You can see this is the kingdom animal, this is kingdom fungi, and this is plants. So there is a common origin for animal and fungi. So they are no more plants, and even you know that they are far apart from plants. Rather, they are close to animals, means they are close to us than plants. Though the most recently published classification divides the kingdom into this kingdom fungi into 7 phyla, 10 subphyla, 36 classes, 12 subclasses, 140 orders, 560 families, 8,600 genera, and over 1,55,000 species. The true fungi which make up this monophyletic clade called kingdom fungi, that I have already told that they have 7 phyla, and this is the latest classification which was given by Hibet et al. In 2007, and it was agreed in the latest dictionary of the fungi, which was published in 2008. That is the last version of the dictionary of the fungi, which is the 10th edition of this book, where this classification was accepted. So there are 7 phyla, as I have already told: Chytridiomycota, Blastocladiomycota, so Neocalliomastygomycota, Microsporidia, Glomeromycota, and Asco and Basidiomycota. So coming to the mushrooms, you know, mushrooms are nothing but the fruiting bodies, or rather you can say fruits of fungi, which are visible through naked eyes and represent a small part of the huge kingdom fungi. So these are the characteristic features, as I have already told: a monophyletic clade being sister to animal kingdom. They are heterotrophic. They can't produce food like plants. So how they run their life? Either they are parasitic, or saprophytic, or they lead symbiotic mode of nutrition. Do you know what is parasite? Any idea? Please tell us. Please. Yes, correct. Actually, the thing is that your answer should be parasitic, means growing on living organisms, right? And saprophytic, growing on dead decaying substances. And the third one is symbiotic mode, means, yes, mutual relationship, going for a mutual relationship with other organisms, right? Neither creating any harm. Please be seated. So these are the three modes through which fungi pass their life. They are unable to produce food, but they have to live. So they are completely dependent on their host, right? Just, you have the idea of parasites, you have the idea of saprophytes. Now coming to the symbiotic mode of nutrition, you know that, yes, you can see I am just giving you a diagrammatic view. You know, if it is a tree, if this is a line of the soil, and you know there is the tree, and if you see the understory, then you will find that there are roots and rootlets, okay? And here the mushroom are. So actually what happened, the rootlets of the tree, they are found to be associated with the hyphae or the mycelium of the mushroom, right? And these how these mushrooms are beneficial? Because they are the organism that promote the nutrient and water uptake. They help in nutrient and water uptake of the plant, and in turn they take food directly from the plant. Hope now it is clear that why they are in mutual relationship, right? Today, if you go to the Himalaya, you will find that luxuriant forests of coniferous and deciduous trees. But you know if you want to know the reason, you know that the reason is the presence of these mushrooms. If you cut those mushrooms, definitely we are going to lose this forest. In turn, on the other way, if you cut those plants, definitely we are going to lose these mushrooms. They are in a mutual relationship which is called as mycorrhiza, right? So coming back to my last slide, they are spore-bearing achlorophyllous, as I have already told, cell wall normally of chitin, and mode of reproduction sexual and asexual. Now coming to the mushroom, you know that this is the classification as I have already told, and these are two highest phyla, that is Ascomycota and Basidiomycota, and most of the mushrooms, they belong to these two highest phyla. Either they are Ascomycota or they are Basidiomycota. Now coming, you know that how the number of, now just let us see how the number of fungi increases with time. You know, in 1943, the first edition of the Dictionary of Fungi was published, right? And it was published with 38,000 species. Again in 2008, you know that the 10th edition of the same book was published with 98,000 species. So the number of species in fungi increased by, we can say, 60,000 in 65 years, right? Again, very recently, in 2023, it was published by Royal Botanic Garden Kew that the present day we have 1,55,000 discovered species in fungi, right? So again, the number of species of fungi increases by 57,000 in recent 15 years. So see, 60,000 in 65 years, and again 57,000 in only 15 years. So with this data, you can realize how the number of fungi is growing, how the discovery of species are coming up, how fast they are going to be discovered till date. And see the estimation. You know that in case of angiosperms, the estimated number of species is 4,50,000. Let us see where we are. We have managed to discover nearly 4 lakh species. Only 50,000, we are near only 50,000 short of our estimation, right? But in case of fungi, see, the estimation is 2.5 million. 2.5 million, and see where we are. We managed to discover only 1,55,000 species. It means approximately only 6.2% of fungal flora or microbiota is known, and we do not know 94% of the fungi, means we almost know nothing. See where we are. This is the latest book published by Royal Botanic Garden Kew, and you know that you can see this is the data of 23, and you can see this is the case of these are the invertebrates, and this is the scenario of the fungi. 2.5 million species are the estimation, and we managed to discover only 1,55,000 species, whereas in case of plants, see the data, and in case of higher animals, vertebrates. So just to give you the scenario, we can say nearly 1.55 lakh fungi are discovered till date. Expected number of wealth is more than 25 lakh. So therefore, we can roughly say that another 23.5 lakh species has to be discovered. Do you agree with me? Presently, the rate of discovery is around 2,350 species per year from the world. Right? So approximately over 1,000 years are required to discover the fungal wealth exists in the earth with the existing manpower. It will take another 1,000 years to discover the species what we estimate, right? If we want to expedite the process, if we want a fast result, so suppose if we want to expedite the process and want to do it by 100 years, then we have to escalate our manpower up to 10 times of the existing strength, which is actually impossible. You know, unfortunately, taxonomies in general and the fungal taxonomies in particular are decreasing day by day, and the species are becoming extinct at a very fast rate due to several anthropogenic activities. And I am afraid that I think that so many species will be vanished or will become extinct before they are discovered. So this is the very pity scenario. Now let us see the diversity of these wild mushrooms. You know, based on my research experience and field experience in different parts of India as well as abroad, I classify these wild mushrooms into 16 broad groups. Let us see the enormous diversity among these wild mushrooms in a nutshell. These are the morphology based 16 broad groups that I am going to share with you. The first one is a gill mushroom, which is, I believe, that is common to all of you, even in the market, even in the forest, everywhere you will find them. You know that these gills are nothing but the fertile structure that bear basidia and basidiospores, right? This is, you know that this is the deadly poisonous mushroom Amanita phalloides, which is considered to be world's most poisonous mushroom, which has amatoxin. When I was in Slovakia and surveying Slovakian forest during 2014, that time I collected from Slovakia, and you know that there are two other mushrooms, Tricholamopsis rutilans, and this is Rusula luteotacta. Just to give you an idea how large they may be, see, my friend Pablo, when he was just coming from the Brazilian forest, he collected this huge mushroom, which is Macrocybe titans, and on my right hand you can see that this is Termitomyces titanicus, one of the world's largest edible mushroom, which is found in Central Asia. And you know that the one mushroom may be of 1.5 meter diameter, right? Which is sufficient for 10 to 15 families, even. Next group, coming to the fleshy poroid, just follow this tag. Coming to the next group, that is fleshy poroid mushrooms. Here you know that the fertile surface are nothing but the collection of tubes ended with pores. You can see the pores, numerous pores are there, and through these pores, basidiospores are dispersed, right? They are called as bollets or bolletoid mushrooms, and there are several groups inside these bolletoid mushrooms. Presently, I am working on bolletoid mushrooms of India. Again, to give you the idea, yes, this is Rugi bolletus. This is one species, Rugi bolletus extreme orientalis. When I was collecting this mushroom from North Sikkim in 2013, and on the right, one of my old friend, Dr. Haribhat, who used to accompany me in several forests of Maharashtra, Goa, and Karnataka during 2006 to 7. Though he was a virologist, but he was so enthusiastic, and he used to accompany me in different forests. So, coming to the next group, that is club fungi. See the enormous diversity, and sometimes, you know, it is very difficult to believe that they are also fungi and they are also mushroom, right? So this is the next group, that is club fungi. So the next one is bracket fungi or woody poroid fungi. They are also very common. You can find growing on different living or dead trees. This is Fomes, this is Fometopsis, etc. and so on. Coming to the next group, chanterelles and trumpets. You know, we have seen gilt fungi, right? Remember what we have seen? We have seen gilt fungi. We have seen fleshy poroid fungi. Then we have again seen woody poroid fungi. And coming to the next group, that is pseudo gilt fungi. They are not the true gills, but instead of true gills, they have veins, rather you can say, or you can say foldings or the veins, the surface, see the fertile surface. Either they are smooth, as in case of trumpets, that is craterellus, or they are with series of vein-like structure or foldings. That is why they are called as pseudogills. And all the photographs what you are seeing today, these are taken by me in different times and different part of the world. So see, these are the, this is, you know, that the chanterelles are highly edible. This is my basket when in Slovakia, and you know that these trumpets, these chanterelles and trumpets, means this one and this one, they actually were given in our food after preparing them, right? When in Slovakia. And see, this is another species of chanterelles, and this is the species of gomphus, and now they are called as turbinellas. Coming to the next group, tooth fungi. We have seen gills, we have seen pseudogills, have seen pores, seen woody poroid fungi. Now the different group, that is teeth. Instead of gills or instead of pores, now we have tooth or the spine-like structure. They are called as tooth fungi. They are hericium. This is Hericium eunthungens. It was discovered by me in 2014, and it is a highly edible species and recommended and edible as well as delicious. So this is the tooth fungi. Then coming to the next group, they are stomach fungi or puffballs. You know that the fungi that we have seen earlier, they all are gymnocarpic. Do you know what is gymnocarpic? Means spores are developed open and they are dispersed automatically, whereas in this group the spores remain covered inside the stomach-like structure, and once it is matured, then the spore is developed and the spores come out. That is why they are called as angiocarpic mushroom. These are the only angiocarpic mushroom found in the world. They are stomach fungi, puffballs or asters, right? This is, this is Lycoperdon, this is Metromyces, this is Geostrum, so this is Scleroderma, etc. The next group, coral fungus. This is, you can see the structure itself suggests that the similarity with the corals, and that is why they are called as coral fungi. They mainly the genera represent, the representative genera are Ramaria, Clavaria, etc. and so on. This is Ramaria subalpina. It was collected by me in 2012, and then later on it was discovered as a new species. Coming to the next group, bird's nest fungi. You know that from the name you can see that that they are very close to the nest of a bird, right? See, these are the eggs, or you can say the peridioles. Peridioles are so nicely placed inside the peridium that the entire structure gives the look of a nest of a bird. That is why they are called as bird nest fungi. There are several genera like Nidula, like Cyathus, Crucibulum, etc. and so on. This is one Nidula singbensis which was discovered by me and Rilin from China in 2014. This is jelly fungi. The structure, the texture itself suggests that the entire thing is very much jelly-like. They are different. There are different species of Tremella. Tremella, maybe Tremella. This is Tremella foliacea. This is Tremella mesenterica, etc., and so on. So just see how diverse the mushrooms are. More I survey, more I explore the forest, more I realize that how diverse they are and how little I know. Coming to the next group, this is stinkhorns. You know that, do you know what is stink? The word stink means? You know, during the development of the spores, these olive color, the texture is covered by olive color mass, and these are nothing but the collection of spores. So during the development or the maturity of the spores, these fungus, they produce foul smell throughout the forest. That is why they are called as stink fungi. Right? And with this unique smell, you can predict, or you can assume that yes, your fungus is there maybe 10 to 20 meters away from you. So they are either phallus or nidula. The next group is cup and saucer fungi. The structure itself suggests that they are cup-like or saucer-like, and there are different species of Piziza, Scutellina, Scutellata, etc. And this is Chlorociboria. Coming to the next group, earth tongues or jelly babies. You know that, see the structure, they are quite similar to the tongue of a human being. That is why they are called as earth tongue. They are different species of geoglossum and trichoglossum. And on your right side, they are also jelly-like, and they also they belong to Ascomycota, and they belong to the genus Liosia. They are called as jelly babies. Coming to the next group, saddles. Do you know what is saddle? If you know, please tell. Saddle is nothing but the seat on the back of a horse. Right? See, the cap of this mushroom, they are so similar with the saddle, that is the seat on the back of a horse. They are called as saddles. They are different species of Gyromitra, Helvella, etc., and so on. Coming to the next group, carbon and cushion fungi. You know, they are stone-like, dead hard, and you know, after maturity, they turn black because of the production of the ascospores. They are different species of Daldenia. This is Daldenia concentrica. These are Xylaria. They are also commonly called as dead man's finger. And rest of the mushrooms, I put them in two broad groups without giving them any special names. So this is one, the reason behind, they are jelly-like, but they are with tooth. So jelly plus tooth, the combination of both jelly and tooth, they are pseudohydnum, number one. Number two, they are spatula-like. The genus is Padularia. And the third one, see how this is different. It is cup-like, right? But see, one-sided open. The cup is one-sided open. If you want to put some water, then the water will come out. So this is not complete cup. They are Oetidia. And the next one, this is one of the most costliest mushroom of the world, or you can say India. This is morels. They are actually belonging to the genus Morchella. And this is the final group. This is cauliflower fungi, Sparacis. This is again shaving brush fungi. This is ear fungi, and this is crust fungi, Piniophora. So this is all about the diversity of wild mushrooms which I have come across during last 25 years. Now coming to the next part, that what basically we do. During these years, I have managed to survey more than 100 forests, either in India or in US or in Slovakia or in Spain. So what we do extensive, intensive, repetitive and periodic survey of forests. Why? That extensive survey is required to complete all the areas. Intensive survey, why is required? Because that the fungus are host dependent. Don't forget that fungus are host dependent. If you have to search those fungus, you have to go for the host. You have to find your host, right? There are several hosts. There are several fungi which are very specific to the host. If you know your host, you will definitely find your fungus. Otherwise, if you do not know what you have to survey, then in the forest, maybe you have to spend lot of unfruitful time and your effort without getting any fungus, right? And repetitive survey is very much required, and periodic survey as well it is required, because the fungi which grow in the early monsoon, they will never come in the late monsoon. So if you cover the whole rainy season, then you can go to note down or to record the whole mushroom flora of a particular area, right? The second point is meaningful collection of young to mature Basidiomata or Ascomata. That we have in the forest, we get young to mature fruiting bodies, or sometimes only mature. So what we have to do, if we have to know a species, if we have to identify a species, if we have to discover a species, then we have to collect their young to mature fruiting bodies so that we can describe the whole species starting from their young to their mature stage, right? And we the taxonomists, or the worker who are working on the systematics or the morphology of mushrooms, we are looking for the more and more parameters. More the parameters you know, more easier for you to identify the species. If you, in the field, in the field when we come across a species, we come back to our base camp, and then we spend around 25 to 30 minutes to work out, to record the morphological features of a single species. And we have to do it in open daylight. Why? Mushrooms are full of colors, right? If you do not do it in daylight, then definitely you will record some wrong colors, which will in turn mislead the whole scientific world. Right? So the most important part is, my most important part, which is marked with red, is thorough and methodical characterization of macro and micro morphology. If you do not do this part, then it is next to impossible for you to identify any single species of mushroom. You have to meticulously observe the characters, record down the morphological features in the field. Then after coming back in the laboratory, we have to take n number of sections from the dry fruiting bodies and to see under microscope to record micromorphological features. So we have one set of macromorphological features which we have to do it in the field or in the base camp, and another set of micromorphological features which we have to do in our laboratory. So when we have the whole set of macro and micromorphological features only, then we will be able to describe a species or identify the species, right? So this is the most important part for any mushroom scientist. Then identification by satisfying your taxonomic ego and the current perspective, because once you have the set of characteristic features, as I have already told, more the parameters more easier for you to identify. So we are always, with the taxonomists, we always want to find more and more morphological features. Sometimes we use chemicals, some chemicals like potassium hydroxide, some ferrous sulfate. Why? Not to know the chemical reaction, but to see if there is any color change. We apply potassium hydroxide on our mushroom, whether there is any color change or not. We apply ferrous sulfate, right? Sometimes we bruise. Why bruise? If we bruise with our finger, then the tissue will be damaged, then the area will be oxidized, and if there is any reaction, and if it causes any color changes, and we will note down, yes, after bruising, it turns blue. I am coming in my later part. So these are the features we have to know, and finally, we go for the documentation to present and uncover the mushroom whale to Government of India. During my journey to the mushroom world, I have surveyed till date more than 100 forests, and we have surveyed several tropical to subtropical to temperate, then subalpine to alpine forest up to cold desert. And based on my experience, I have seen that the most fruitful forests are the temperate to subalpine regions in our country. See, in last year, I was in Dhakuri. that is of Kumaon Himalaya of Uttarakhand. And you can see it is a luxuriant forest of mainly of Quercus semicarpifolia, and we got very good collections of wild mushrooms. Yes, there are n number of moments to share, but you know that the time is very short. Yes, you can see that our team is crossing the last village of India, Mana, and we are approaching to the Bosudhara Falls. Here you can see that my team, we are waiting here. Actually, our tent is packed in the forest, and we are waiting for few porters. We will come from 10 kilometers apart. And here, yes, my team, we are at the Indo-Nepal border at Chiavanjang. Yes, here you can see that we are crossing a coniferous forest in Dafiadura Pass. And you know that how we take the survey. You know, it is not that we are visiting forests, our mushrooms are coming towards us. This is not that. You know that we, up to some part, we go by vehicle, and then from that point, we have to take long trekking tour, long trek, deep into the forest, and we put our camp, we set our camp, and inside the tent we have to stay 4 to 5 days or 6 days. We try to cover the surrounding forest as much as possible, and once we feel that the area is almost covered, then we pack our tent, and then we move to the next forest by trekking another long trek. You know, sometimes we don't have sufficient food, even sometimes we don't have sufficient water. But students, one thing I can say you that, you know, that once you will have the joy of discovery, you will forget all the pain and agony you have in the field. That I can make sure. So this is one, you can see this is in the year 2001 when I was a student like you, and yes, of course I was doing my PhD, and the entire team was in Dafiadura forest, which is full of you know, tiger, leopards, and wild boars. It was very dangerous. The forest was itself. Here, my camp in Chokha, in North Sikkim, in Kanchenjunga Biosphere Reserve. And here, my camp at Thalisan. Here you can see our camp in Majharatal Forest. This is a very recent tour. And now coming to the photography, how we take good photographs. You know that we have to go down to the object, first of all. Secondly, you know that we do not use the flash of the camera. Instead, we use, you know, we use this thing so that we can get the light, but to avoid the flashlight of the camera. Here, you can see another one interesting thing. This is a tripod, and our tripod is differently modified. In the extended part is going below up to the specimen. These are my you know that these are the things which I carry in the field. As I already told that after collection, the most important part is to undertake the macrofungal survey or to collect the morphological features of every mushroom. So you can find me sometimes in the storeroom, sometimes in the forest with students, sometimes in the veranda, because you know that very soon I come back I start early in the morning by 7 o'clock or to 7:30, and I try to come back to my base camp by 1:30 to 2. Then then as I already told that I have to finish my macromorphological workout in the daylight only. So time is very short and we have to finish all those works. And we usually use this type of format. We have to record different features of the pileus, that is the cap of a mushroom. It is, you can see the measurement, then the diameter, the color, etc. Similarly, we have to note down the cuticle, that is the upper surface. The features of the cuticle are very important, whether they are viscid or glutinous or moist, etc. The marginal characters are very important. Color is also very important feature. And then the lamellae, that is the gills. We have to note down the features like how the gills are attached to the stipe. This is very important feature. How the gills are attached to the stipe or the stem of a mushroom, right? So this thing we have to record. Then the density of the gill. So this is also, these two are very, very important part for us. And then the stipe, the shape, size of the stipe, then if the vulva present, then we have to record. Then we have to note down the texture, color of the flesh or the context. If it has latex, then we have to note down the color of the latex. And then we have to take, sometimes we have to take the taste. That is also important feature. And the odor we have to record. We have to take the spore print. We cut the pileus or the cap and then put it on a black or the white piece of paper, and we keep it for the whole night. And in the next morning, when spores are deposited on the paper, then we have to record the color of the spore print. This is also very important. Black spore, probably it is coprinus. White spore, probably it is marasmias, probably it is rusula, etc. and so many. So we can reach up to genus level sometimes, right? So every morphological features are very important for us. And remember that we have to gather, we have to note down, we have to record the whole set of morphological features of a single species, right? See how the spore prints are, are taken. You know that this is a pileus of a boletus, and it is kept. And in the next morning, you will find this type of olive spore print on your white piece of paper, right? And this is the identifying feature of this genus. See, as I have already told, that several mushrooms, they ooze latex. Latex are also very much important, and they are, that sometimes significant. They play a significant role in taxonomy. See, you can find the almost transparent latex. Sometimes the latex are yellow, latex are orange, or transparent to semi-transparent. Then see the latex turning the gills violet. That is also a characteristic feature of some species. See, here the orange latex is coming out from Lactarius deliciosus. So these are the features we have to note down as well. As I have already told, that while noting down the pileus morphology or the cap morphology, we have to see how these hairs are arranged. Maybe velutinous, hespid, then pubescent, etc., and so on. Maybe with squamules, as it is often we find in agaricus. Similarly, just to give you an example, I was working on a particular group of mushrooms, and I can see these many types of pileous surface. See, they are either smooth to slightly rough, they are subvelvety maybe rugose or with convoluted pileus, maybe reticulated, right? Maybe squamules with squamulos, maybe flocos, or maybe pulverulent, means covered with powdery substances, maybe cracked or areolate, or maybe with huge gluten. See, it was so glutinous. This was the species, this is Astrobolatus olivaceae glutinus. It was discovered by me and published in Q Bulletin in 2014. And see, the species was so glutinous that always I collected with the dead flies stuck on it. Right? So, again, that as I have already told, that the pileous margin are also very much significant, and they bear some special characteristic features. Maybe enrolled like this, maybe incurved, maybe decurved, maybe plain, or uplifted, or with appendages. See how distinctly appendages they are. As I have already told again, that that the attachment of the gills are very important for any gilled mushroom, right? We have to record the attachment pattern. Either they are free, see, it is not attached to the stipe, so we can say the gill is free. Maybe adnexed, means sharply attached, maybe sinuate or notched, maybe broadly attached, that is adnate, or going down, that is decurrent or subdecurrent. I have told that the density of gills are another important parameter. The gills may be distant like this, see, it is present in Lactarius Ocrogalactus. Then see, maybe subdistant, this is distant, this is subdistant, and this is crowded, as it is present in Lactarius piperotus, right? So all are the important morphological features we have to record in the base camp by the same day after your collection. Coming to the stipe, as I have already shown, see, the stipe may be of different pattern, maybe cylindrical, maybe centrally broadened, maybe with bulbous base, or maybe abruptly bulbous base. Now see, I was working in a different group, and I got this type of stipes. That they may be this clavate, or the surface may be with scabbers. See, you can see the stipes with scabbers. Stipes may be with dotted to glandular dotted, maybe with faintly reticulation. You can see the stipe with faint reticulation, then with strongly ridged to reticulate. Again, the same thing. This is found in Retiboletus. And see, remember the species I have already shown you with thick gluten and the flies stuck on it, and see the stipe surface. So this is lacerate. This type of stipe surface is called as lacerate. And then this is flocose, as in case of Strobylomyces mirandus. And this is winged. See, the surface is with high ridges and wings. So again, reticulation. As I have already mentioned, that the spore print color is very much meaningful for us. If we have black spore print, then we can say quickly that yes, maybe our species is either Coprinus or Coprinellus or etc. If we have yellow spore print, then we can say yes, maybe our species belong to the genus Rusula. If we have purple brown, then we can say yes, it may be purple brown to black, maybe our species is Agaricus or so. So this type of morphological features will help you to go somewhere, right? But it doesn't mean that they will give you the final indication or will help you finally. No, we have to go for the whole set of features, out of which this is one of them. These are the drawings that I used to do by sitting inside the tent or in the forest, because you know that if you come back from the forest, then once your specimen is dry, then the shape, we are going to lose the shape, right? So we have to finish this workout, we have to draw the species, to record the shape in the field itself. And these are the, you can see these are my drawing on to show the attachment of the gills from the mushrooms I collected. These drawings are made inside the forest or in the base camp. And see, these are the dryer that I use in the field, artificial dryer. You know that sometimes we use kerosene oil, or sometimes we use electric bulb to dry them. Once the workout is over, the next thing is that we have to go for overnight drying. Because you know that our target is we have to bring the species, these specimens, to our laboratory to see the micromorphological features as well. We have to take n number of sections from the dry specimen, and we have to observe under microscope to see the micromorphological features, right? So that is why we have to go for drying. And coming to in the laboratory, if we take the sections, you know that these are the hyphal arrangement, maybe interwoven like these, maybe divergent. See, divergent, the hyphae are running away, going down and running away, they are divergent. And see, this is convergent. Again, you know, coming to the micromorphologically, they are also very important feature like basidia and cystidia. You have heard about basidia, right? Do you know about cystidia? Cystidia are the structure which are found along with basidia. They are also present on the fertile surface, but they are the sterile structures and very much characteristic for the identification of fungi, right? So we have to take the sections. We have to see how the cystidia looks like. Now, you know that the cystidia maybe they are pileocystidia, means when cystidia are present on the cap surface of a mushroom, they are called as pileocystidia. When they are present on the stipe surface or the stem surface, you can say, they are called as caulocystidia. But we have to observe the cystidia, right? See, these are my drawings, micromorphological drawings. These are the different patterns of cystidia I observed. Maybe they are thick-walled, maybe with appendages or tails. They are called as laginiform. They are, see, this is moniliform cystidia. These are, fusoid, or, the capitate to subcapitate. And these are the basidiospores which I have observed. They may be minutely warted, maybe spinoid, or maybe winged. These are my drawings based on the species I have gathered or which I have worked out under microscope, right? Maybe they are with zebroid pattern, maybe with a complete reticulation, etc., and so on. Yes, again, I was just 4 to 5 years back, I was also working on a different group called as Rusulesi, and where I got these many patterns of spores, spore ornamentation. So spore ornamentation is also a very important feature, micromorphological features, which we have to record and note down and compare with the allied species to identify of the species of our interest. Right? These are my drawing with the maybe with camera lucida or with the drawing attachment tube. Now, once we have the macro and micro morphological features, see, we have started working from the field. We came to the base camp. We recorded the macro morphological features. Then we came back to our laboratory. We have the micro morphological features. Now we have the combination of macro and micro, right? So once our workout is over with macro and micro, say we have set of 70 to 80 characters, then we have to prepare this type of illustration. These are the you know that these are the drawing, as I have already told, which I prepared in my base camp in the forest, and these are the micromorphological drawing which I prepared in the laboratory. So this is the whole drawing for a single species, and this is also the whole drawing for this species, which was published in the renowned journal Mycotaxon in 2004. We discovered this species with these macro and micromorphological features. Now coming to the pigmentation, you know that mushrooms inspired the cuisine in many cultures. That is quite known to you, right? And many are used in folk medicines, right? But it is little known that they were used as textile dyes as well. And they have the vast range of colors or pigments. They don't have chlorophyll, they don't have anthocyanin, but what they have? They have carotenoids, they have terpenoids, they have quinones, they have sesquiterpenoids, palvinic acid, etc., and so on. And why we are looking for these pigments? We, the taxonomists, we are looking for different kind of morphological features. And you know, these pigments, if you see some species, this color changes with maturity. Once the species becomes mature, the color of the fruiting body changes from one color to another, right? Some are changing with bruising, as I have already told. If you touch your species with your finger, immediately the color will change. And we, the taxonomists, we have to note down this change. Why? To increase one more parameter, which is unique for our species, right? So, just to give you an example, see, remember this is fleshy poroid mushroom. We don't have gills here. We have pores, right? And you know that you can see this is the impression of my fingers in the forest. And once the pore surface was yellow, bright yellow, I just touched it and immediately it turned blue. And this was my character. I am a taxonomist. I have to observe those features because I have to separate my species from others. Right? So this is one species. Why this color change? If you ask me why this color change, then I have to give you the breakup. You know that they are boletes. They have variegatic acid and xerochromic acid. And once they are oxidized, these variegatic acid and xerochromic acid, they are converted to their blue chinon methid anions. And that is why it turns blue. Similarly, you know that this is a species, Lactaria subindigo, that oozes blue latex. which is very distinct in the field. This is the world's most beautiful fungi, that is Amanita muscaria. It was my collection from Slovakian forest as well, because in India we don't have Amanita muscaria. We have its close allies, but for sure we don't have Amanita muscaria, right? And so here, you know, this color actually, the combination of three colors, purple beta cyanine, then orange beta xanthine, and yellow musca flavin. So combination of three pigments made this beautiful color. I collected one species of Agaricus xanthodermus. You know that these are my finger impression only. It was white. I touched, immediately it turned yellow. It had leucophenol that produced, when it was oxidized, it produced yellow agaricone, and immediately it turned to yellow. And these were my parameters to separate my species from the rest, right? You know that, will you believe that they all are same species? Of different pigmentation, right? That means pigmentation is not the final word. As I have already told, that we have to gather, we have to record the whole set of features, right? Do you also believe that all are different species with same pigmentation, right? Here your taxonomy work. Here is the reason for paying lot of attention, to gather lot of, to put lot of effort to note down all the macro and micro morphological features, right? So problem, now coming to the problems in identification. Among the species in several genera, we have seen that there are a large number of overlapping characters. Do you agree or not? Right? Often the delimitation means separation in species are mainly based on the variations that exist in cell level, and we have to take n number of sections and to see under microscope to record those parameters. It is quite difficult. How you will separate them in the field? So it is very difficult and it is tedious. It is a tedious job, right? So this is another problem. So many genera have cryptic species or species complex. Do you know what is cryptic species? Have you ever heard? Cryptic species means similar looking different species. All look similar, but they are actually different. How did we come to know that they are different? Morphology tells they are same. And that is why, since last 200 years, we call them as a single species, maybe Lactiflus volemus. And you know that just to give you a history, no, I will come back later anyways. So just remember this, similar looking different species is called as cryptic species, or species, the whole group of species, similar looking that species is called as species complex, right? Intercontinental lookalikes are quite common. As I have already told, that in fungi, the species complex, we have n number of species complex and similar looking species. Maybe we have a similar species of Asia with Africa. Maybe we have we have 2 to 3 species which are very close to the European samples. So, but you know that when we go for morphology, we think that yes, they are same. So that is why we have to go for the whole set of morphological features. Along with, we have to go for molecular analysis. And when we study in combination of morphology as well as molecular phylogeny by extracting the separate genes, separate barcoding regions, and when we have analyzed the sequence data, and then when we prepare their phylogenetic estimation, then we realize, no, yes, our species is distinct. Our Asian species is distinct from the European or the North American or the African, right? So this was the reason that in 2004, first I started working on molecular phylogeny. You know that that time when I started, no one in India worked with molecular phylogeny. And you know that I went to nucleic acid exploration facility in US, and then I was trained by the US people. And then you know that for the first time in India, we started working with molecular phylogeny in 2004. And then from that, so what is the reason that I have already told? Morphology was not at all sufficient for any fungi. Now it is well established, right? So many traditionally used, these are the problems that we faced, and that is why we started working on molecular phylogeny. Many traditionally used phenotypic characters in fungi do not necessarily predict their phylogenetic relatedness, right? Number one. Number two, multiple morphology-based species bear same DNA. Maybe morphology is different. Maybe, as I have already shown you, that the pigmentation is different, but they are same species, right? So, third thing, multiple cryptic species are called under a same name. As I have given you an example, that the Lactiflus volemus is found in Europe, Africa, Asia, everywhere, North America, all started calling as Lactiflus volemus, right? So these are the reason. And finally, you know, and now we know that 1 or 2 genes even are not sufficient for fungi. Instead, we have to follow the current gold standard, that is concordance of multi-gene genealogy. We have to consider at least 4 to 5 genes from a single species to identify your species with morphology and molecular phylogeny, right? So the equation is changed. Earlier it was based on only field observation we used to identify. Now, you know, after that, the time came when we considered both macromorphology as well as micromorphology. Then we started working on macromorphology, micromorphology, and based on the literature. Why we have to search literature? Because so many countries started publishing species. Then we have to go for the literature. We have to see where our species stands, who are the allied species, and where our species differs from others, right? And finally, the equation is changed, that we have to now go for molecular phylogeny must for every species to identify your fungi. So in Botanical Survey of India, we basically do to uncover the mushroom diversity or the hidden mushroom treasure. We do basically the 4 things. We do discover the novel species that is new to science, or we do discover new genera which are new to science. We resolve intercontinental conspecificity. Hope you understand what is intercontinental conspecificity. Same name called by Europe, same name called by Africa, same name called by Australia, same name called by USA, right? They are intercontinental conspecificity, means same species called by so-called mycologists, they called them as the same species, but they are not like that. So we resolve this intercontinental conspecificity, and finally we report the new record for Indian microbiota. These are the 4 things. Now, finally, I will just to show you few small stories very quickly. You know that just to show you at a glance how we solved several problems with morphology as well as molecular phylogenetic data. Right? You know that I did my, fortunately or unfortunately, I did my PhD on the genus Rusula from India. And you know that in this genus we have around 2,300 species. And out of these 2,300 species, more than 850 species are of this coloration, means red. It is next to impossible for anyone in the world to separate them in the field. No, it is not possible. In the field, if I give you 850 species, how you will separate them? This is not possible. What do? We, as I already told, we record all the macromorphological features. Then we, coming back in the laboratory, we record all the micromorphological features. You can see from my drawing, these are cystidia, as I have already told. Cystidia are quite small. We take the sections from the pilia surface. This is the surface or the cuticle, and after taking the section, we can see this type of structures, which is quite unique for my species, right? Then we have to go for the spore ornamentation, whether it is reticulate or it is spinoid, it is minutely worded, or the incomplete with incomplete reticulation, etc., and so on. Finally, yes, when your species is completely worked out, then we went for molecular phylogeny. Here, you know that, as I remember, I took only 2 barcoding genes. One is the ITS and LSU, and based on our molecular phylogeny, yes, you can see that the red mark species is my species, which is separated from all the sister species from all over the world. These are the phylogenetic estimation of the particular group of Rusula which are present all over the world, right? Maybe my species is very close to another species which are found from US or from UK, right? So similarly, you know that, remember that species. This is Astroboletus olivaceoglutinus, and it was published in Q Bulletin, and you know that the macromorphological features, you can see these are the spore ornamentation. See, the spores are how nicely speated. This is the scanning electron microscopy of the spore, and you can observe that the spores are nicely peated, and these are the other structure of the pilipelis in the spore. And from my drawing, you can see the cystidia are different, and the basidia are of 2 types. So these all the morphological features, but yes, we were not satisfied, and not the reviewers will also satisfy of the will be satisfied. So you know that we went for, though our species was unique, but still we went for molecular phylogeny. We took the sequence data, and then we ran the molecular phylogeny, and then it quickly comes apart from all the known species of Astroboletus. Coming to the discovery of another species, very interesting story. You know that the Suillus laricephalus, you know that the name itself suggests that phylus means loving. Laricephalus comes from, it grows under larix. Have you heard about larix? Larix is one of the coniferous trees which are found in few pockets in India. I was surveying North Sikkim, and there we have a good population of larix, and you know that this species was collected under larix, right? I did my complete workout. These are my drawings. These are the cystidia. Here the most important point is the collection of tuft of cystidia are present over here, which is very unique for this species. But you know that before I go for identification, I went for the molecular phylogeny, and then what I found, yes, my species come very close to another species which was 30 years back someone worked on it from Nepal. It was very difficult for me, and I tried a lot to find out that person. Fortunately, through my few friends in America, I found that Dr. Carter. He was so old, but when he came to know that my species is the same one, then the very next day, he sent his whole thesis, which was written in 1982. He sent his whole thesis to me. Yes, Dr. Das, please go for it and let us publish it. Then finally, we published this species with the help of morphology and molecular phylogeny, along with Dr. Cotter from Duke University in cryptogamy mycology in 2014. And see, what is the most important feature here is that, you know that these clad bearing 1, 2, 3, 4, 5, 6, and 7 species, and all are larynx loving. These species only grow under larynx. Remember ectomycorrhizal association? I have told that there are several species which are very specific to the host. They only know larynx. If larynx are not there, they will never grow. Right? So all larynx loving species, they have the same genetic makeup and they cluster together. And we managed to discover this species. You know that we discovered the first Asian species in Rusula subgenera Archaea. In this group, there were no species discovered till date, and we were the first person to discover it. There were species from US, there were species from Europe, but we were the first to discover from India. Me along with my teacher, Dr. Bart Bayek. So we discovered this. Yes, finally I will tell this story in a nutshell. You know that I have already told you about cryptic species or the species complex, right? This is the species, world-renowned species Lactiflus volemus. Lactiflus volemus was discovered more than 110 years back from Europe, right? Because they are so advanced. Even then we didn't start our mycological workout. But that time it was discovered. It was discovered by Parsoon. And then gradually what happened, the similar looking species. Remember, cryptic species means similar looking different species, right? So similar looking species started to be discovered. Some people found from Asia, some people found from Europe, North America, Australia. But unfortunately, the name of Lactiflus volemus was misapplied to all those similar looking species. Everyone told, yes, we have Lactiflus volemus, right? And they reported in their official record. So we knew that, yes, Asia have Lactiflus volemus, Europe already have, US already have, Australia have, so everyone have Lactiflus volemus. This was the scenario of Lactiflus volemus. In 2012 only, you know that, yes, even you know that myself while doing my PhD, I also called them as Lactiflus volemus from Asia, from India, right? Okay. But in 2012, when we got all those species, you can say that the nearly 25 to 30 species of similar looking, then we started rethinking the whole scenario. And then what happened? We go thoroughly for the morphology. But we realize that the morphology enough is insufficient to say that yes, Asian species are different, right? Now, see, we went for the scanning electron microscopy of all the spores collected from different part of the world. We found, yes, all the spores are of same looking. Morphology fails, right? We were puzzled. I collected 1, 2, 3, 4 species of similar looking, right? I placed them as Lactiflus volemus. So this was a very pity situation. And then finally, yes, I went for the thorough characterization. This was the macromorphology. These are the micromorphology. And the most important character was thick-walled cystidia, long thick-walled cystidia. See the wall thickness. They are so thick. And the spores are similar. You can't separate. See, this is for the first, second, and third species I am showing you. This is the first, this is the second, and this is the third. Almost similar. See, they have thick-walled cystidia. They also, and they also, right? So what are the difference? Yes, there are minute difference. See, in case of Leptomeras, they have very slender stipe, right? It is quite gigantic. So with these features, with these morphological differentiation, with this minute difference, can we establish them as a different species? No one will believe. They will throw our proposal in the dustbin, right? Yes, we did. Well, no, you go out. We went for, then we took the sections from the Pilias cap, and see, we got another difference. In case of the last species, that is Perciformis, we have long hairs, see, on the Pilias surface. Whereas in case of the first and two, that is Digitas and Leptomeras, we got small cells. This is also insufficient. They can say, no, this is the range of Lactiflus volemus. You can get from small cell to large cell, but they are Lactiflus volemus. They will never believe. Okay. Then we go for 4-gene molecular phylogeny. And then we show the world that, see, this is the original Lactiflus volemus from Europe. This I got from European collection. I procured the species from Paris and from Switzerland. And then also I analyzed them, as well as I analyzed the Indian species and see where they stand. These are the European species of original Lactiflus volemus, means Lactiflus volemus sensu stricto, in strict sense. Indian species are far apart. There are 3 more species inside, and here are the first Indian species, second Indian species, and third Indian species. Then we finally, and again, while working with the 4th species, I prepared another phylogenetic tree, and then I showed them, yes, now we know that the Lactiflus volemus is no more a single species. It is a complex of 22 species from the world. 7 to 8 are from Asia, and rest are from different part of the world. And now, because of our publication in cryptogame mycology as well as fungal diversity, now we know that yes, the Lactiflus volemus is a complex. They are no more a single species. Now, similarly, you know that we differentiated. Earlier we know that this is also Lactiflus Lactarius chrysorius. Now we established that yes, no, we don't have in India Lactarius chrysorius, but we have Indochrysarius, another new species we published. So yes, these are the differences. I am not going so details. Yes, another story, small. You know that this is the species that, this is another genus, not Lactarius, but Rusula. Here you know that if you touch it, it will turn brown immediately. So this is the characteristic feature. You know that when young, first it will be whitish orange, then it will turn dark orange brown, and if you touch the gill, then it will turn orange. This is the characteristic feature of Rusula compacta, which was discovered in 1912 from US. But we the mycologists from Asia, Europe, everyone, we started calling our similar looking species as Rusula compacta, similarly. But morphology here also, see the different, and while working with this Rusula compacta group, I collected 2 species from Himalaya. One is there, yes, one is this one, which I collected from 12,000 feet altitude. And another one, this one, I collected from 6,000 feet altitude. Now the question came to my mind, whether they are same, first question. Second question was, whether all are Rusula compacta, which was discovered 100 years ago from US. So these were the questions. To solve all those issues, we found that yes, there are some differences. Our 12,000 feet collections, here you know that in case of this one, we got forked gill. See, the gills are forked. Here you can see the forcation, branched gill. Here we don't have branched gill. So this is the difference. But with this difference, can you send your proposal? No. No one will exist. No one will listen to you. Then we go for, with classical taxonomy, we go for molecular phylogeny. And finally, we found this is original Rusula compacta, which was established 100 years ago. And then we have from India, we have compactoides and pseudocompacta. This is compactoides, and this is pseudocompacta we discovered. And these are the 2 species which come under Rusula compacta complex or cryptic species, right? These are the drawings, almost same. I am not going detail because it will take time again. Just this is the final story. You know that, could you believe that this puffball-like fungi, they belong to the same genus of these gilled fungi? I was utterly surprised. When my student brought these species to me, then I told, did you do the macromorphological characterization in the field? Then he told, no sir, I didn't manage to complete it. Well, I'm okay. So forget about it. Go next year. So in the next year, he went again. But that time, he put his valuable effort and managed to do the whole characterization. And then one clue only I got, that it is having the latex. See? It is also oozing latex. And I know that there are only in the world, there are only 2 genera, Lactarius and Lactiflas, that oozes latex. Then I realized, yes, this may be something which belong to Lactarius or Lactiflas. It is the allies of these gilled fungi. All the gilled fungi, all the species of Lactarius and Lactiflas are gilled. But here you can see due to the evolutionary pressure, they didn't develop, but they remain like this. And finally, when we did the morphology along with multigene molecular phylogeny, we found, see, this is the tree of the phylogenetic tree of the Lactarius, and our species, Lactarius rajmolensis, take the distinct position among the all known gilled species. And here is the discovery of the first sequestered milk calf from tropical India. And from that day, we came to know that yes, these sequestered fungi may be of the same genus of these gilled fungi. Right? So, morphology, if we have to do morphology, we have to do the whole. It is not that, yes, I have done only 1/3. No, it will not work. Right? These are the drawing. Yes, it was found in Sal forest. It is also available in several parts of Midnapore district. And this was the species which was collected, and these are the fruiting bodies. You can see the latex oozing out, and yes, these are the spore ornamentation. This is my drawing, and finally we published it in Cryptogamy Mycology. This is the new genus. Hope you all understand the differentiation between genus and species, right? Tiger and leopard both belong to Panthera, right? So similarly, but you know that this is the first genus which we discovered on the fleshy poroid fungi, that is Indopora sori, and it was published with the thorough macromorphology along with micromorphology and our multi-gene molecular phylogeny. See, you can see this is our molecular phylogenetic result, and our genus Indoporus, it is distinct from all the known boletoid mushrooms. Here we are talking about the genus level. That is why only the genus names are mentioned, right? Similarly, we discovered another novel genus from our well-known AGC Bosch Indian Botanic Garden. This is Mycorrhaphoides. This genus was discovered by us in 2017 and published in Nordic Journal of Botany with morphology. And see how different this genus is. This is a tooth fungi. You can see the tooth. It has a long central stand with several pili. See, several pili, only a single stand. And it has zonation, zones, and see the multiple pili are arranged in tiers, and the spores are smooth. And see, these are the clamp connections. Multiple clamps are present with the cells, and it was finally discovered with the multi-gene molecular phylogeny. Yes, as I already told, we also publish new records to India. So this is one of them. Do you understand what is new records to India? Means this species is published from other country already, but if you have the same species, then you have to give the proof with morphology as well as molecular phylogeny. See, here our Indian species, yes, this is Cortinarius longistipitatus. It was earlier published from Pakistan. In fact, the original type material, it is published from Pakistan, and now we know that we have also in India that Cortinarius longistipitatus, right? And this is the sequence data from our Indian collections, and these are the sequence data from Pakistani collections. Now you know that due to some unavoidable circumstances, today I can just show you through microscope, but you know that this is our daily work. If you take the section from the gill, and you know there are mainly two things you have to observe. One, basidia, and the second thing is cystidia. As I already told, they are also present on the fertile surface and very characteristic to identify your species, right? So if you take the section as we do every day, either me or my students, every day we are taking sections from the gills or the pores or the tubes, and we get this type of cystidia. You can see this is a cystidia with appendages, and these are normal cystidia that are fusiform or subventricose, or sometimes maybe ventricose rostrate or laginiform, etc., and so on. Similarly, you can see it is quite evident that you can see the basidia as well as cystidia in the fertile surface. So we have to, basidia are not so characteristic, right? In Basidiomycota, basidia are common, right? So they are not so characteristic to us, whereas the cystidae are very much characteristic because they are different in shapes, in sizes, in content, in color, etc. and so on. Right? Similarly, as I already told, that the spores are these are some photographs from from our species. You can see this is a species of Rusula, and if you add Melzer reagent is something where you will find that the starch iodine reaction, if it happens, it will convert the ornamentation into black, and that is why you can say that yes, my spores are ornamented. Whereas on the right, this is a spore of a fleshy poroid fungi, bollets, where the spores are smooth. Yes, as I have already told, this is Pilipelis. Pilipelis means the pilious surface, means cap surface. If you take the section of the cap surface and see the upper layer, this upper layer is called as Pilipelis, right? So if you see the structure, you will find this type of erect to parallel or sometimes submerged under gluten cells, right? Here the pattern itself suggests that they are erect, right? So this type of erect pattern is called as trichoderm. These patterns are very characteristic in different groups of wild mushrooms. If you say that yes, trichoderm is present, then people will understand, yes, probably this person is working on bollets or bolletoid fungi or fleshy poroid fungi because it has trichoderm. If some person say yes, no, it has intermixed cells or parallel or maybe sub-erect cells, then people will think again, no, this is probably it is not bollets. Maybe it is a different group of mushroom, maybe agaricus, maybe cantharellus, etc. and so on. So these are very characteristic. So you know that these are the mushroom families. You know that as I already told that I know very little. So these are the mushroom families that were explored by me since last 25 years. Mainly Rusulaceae, Bolletaceae, Hericiaceae, Gomphidiaceae, Suilaceae, Bondargiaceae, Cortinariaceae, etc. and so on. And we managed to discover 2 novel genera and over 155 new species, that is new to science, from this country. And these, you know that as I already told, yes, you have discovered means you have to publish in different scientific journal, and it will be thoroughly reviewed, it will be grilled like anything, and finally, yes, when they will be bound to give the surety, yes, no, you are working with a new species, then only your species will be published. So these are the journals where I have managed to publish the cryptogamy mycology from France, Mycotaxon, US, fungal diversity, which have present day highest impact factor, that is 20, then phytotaxa, then fungal IMA fungus from Netherlands, Mycologia, then Nova Hedugia, fungal biology, Q bulletin, Cydoia, Mycosphere, etc., Nordic, Turkish Journal of Fungi, Mycokeys from Bulgaria, etc., and so on. Yes, you know that when you have n number of samples on your table and you have to do the workout, it is hardly possible to write the books, right? But still, you can say that I managed to write few books. This one, 100 Wild Mushroom of Slovakia, which was published from Sweden, and then these are few other books, and also some other books which I am not showing here. You know that we the mycologists, we the mushroom scientists, we meet every 4 years in different parts of the world. You know that it is not possible for us to go there every time because there are huge financial matter, then permission. As I am a government servant, I have to take n number of permissions from Government of India to leave the country. So, you know that I managed to go as they invited me. I managed to go in 2014 to Slovakia, while where all my colleagues came from different parts of the world. And you can see that they are so enthusiastic. This is, next to me is Ron Peterson, who was from US. He was, that time he was 78 years old, but he, you know that along with us, every day he went to the forest. He came back with a large number of samples. Throughout the day up to 9 o'clock he used to work. This is the thing which we have to learn. So, yes, this is this is my table in Slovakia, and we are working on, I am along with 2 other mycologists in Slovakian forest. And you know that I had so many samples to observe that I didn't manage to do any other thing in the field because you know that if you see that your table is full with mushrooms, and if you do not know them, then your duty is to complete the workout and to see, because you know that this is the golden opportunity for me to go there and to see European species. So, you know that nothing makes sense in biology if the organism studied is not identified, right, correctly. And misidentification or misnaming has unfortunate consequences, where not only in the accuracy of taxonomy, but also in the domains like agriculture, breeding, conservation biology, ecosystem management, climatology, etc. And yes, we, since last 60 years, we are serving for the country. You can find me, he is my teacher, my supervisor, he is my grand supervisor, he is my great grand supervisor. All we are there in a same seminar in 2006. He is Dr. J.R. Sharma, he is Professor Lakhan Pal, he is Professor K.G. Mukherjee. So we are serving this country since last 6 decades, and now you know that the exploration is still going on, and I am working with my several students. They are some of them are placed, and they have taken up the mushroom flora of different parts of the country. Some are working in Shillong, some are working in Arunachal, some are working in here, some are placed outside. One of my students is there in Thailand. He is working on Thai species. So this was the final slide. You know that myself representing the country in the first world mushroom one, first world seminar, World Conference on Wild Mushroom, which was organized by the Spain government in 2007, and I was invited to deliver the speech on the opening session. So this took place in Cordova, Andalusia in Spain. And this is, I am just focusing my dish of evening. You can see it is full of mushroom. Even the sauce made up of Boletus edulis. They are so crazy. So thank you for your patient hearing and for your presence. Thank you.
Really, sir, we have enriched ourselves. So many information that make me surprised. We are too teeny to learn everything about the diversification of fungi. Please clearly clarify paraphysis, trichogyne, and cystidia. Is there any role,
See.
or there any relation, or they are differ from each other?
No, no. Actually, the thing is that cystidia, they are mainly found in Basidiomycota. That is why I focused, because my focus is mainly on Basidiomycota. And you know that the most of the paraphysis or this type of things you will find in Ascomycota. So actually, it is very difficult to say what are the what the role they are playing, but yes, all are. You rightly asked this question, because all are mainly engaged to disperse, do the dispersal of the basidiospores or ascospores. So these are the main thing, mainly.
Thank you, sir. Sir, till to date, I knew that there are, according to phylogenetic classification, the fungi placed into a several kingdom, that is the mycota, and under this kingdom, we learned 5. That means Chytridomycota, Zygomycota, Glomeromycota, Ascomycota, Basidiomycota. But today I learned another new that also enriched my knowledge.
Yes, if you, sir, actually, the question you asked, it is quite sensitive. You know that the fungal classification still is a state of flux, right? Every now and then you will find the classification is changing. And very recently, in 2023 end, there is a classification came by the Hyde group, Professor Hyde from Thailand, and they have separated the entire kingdom fungi into 20 phyla. Today I didn't focus on that, because that is quite puzzling, and day by day, you know that the more we know about molecular phylogeny, more we face the advancement of the phylogenetic system, then you know that the classification will take a different shape, and maybe something different what we do not know till date. So I have focused only a very simple classification, which was completely agreed by the Dictionary of Fungi latest version, and which was published by the Hibbett et al. 2007, where the kingdom fungi is divided into 7 phyla. And out of this, Ascomycota and Basidiomycota are the highest group of phylum.
Okay, sir.
For the convenience of the gathering.
Really, that is the Russula species identification. It is interesting and surprising to me.
Thank you,
and
sir.
it was a great challenge to you.
Thank you, sir. Thank you so much.
If you have any question, dear student, sir is ready to give your answer. Really, he is a veteran in this, in his branch. Thank you, sir.
Sir, we have collected some fungi. Can you please give some?
Clue?
Yes.
Yes. This is, see, first of all, what I have told you that yes, in the field you have to note down n number of features, and then followed by you have to go for micromorphology, then only you will be able to identify the species, right? So as you have collected, this is Schizophyllum. This is Schizophyllum, and it grows on so many deciduous tree trunk. I am sure that you have also collected from tree trunk.
Yes, sir.
And these species produce Schizophyllum, which is an anti-cancerous drug, right? So this is medicinally important species, right? But yes, it is not possible for me to tell you immediately that what is the species name. I can tell you up to genus, because as I told you that it took lot of exercise we have to do to identify up to species level. So this is another species. Yes, you know that this is so young to tell you something, but still I can say this is a species of Ganoderma, which is also medicinal, right? This is again bracket fungi in my group. My first group was gilt fungi, second group was fleshy poroid fungi, and third group was bracket fungi, which grows like producing a bracket shape on tree trunk. So this is bracket fungi. Again, yes, it is almost damaged. I can see nothing, but still as the thing is black, I can say that maybe that your species is Coprinus or something, because it you know that it produces black spores. So this one, right? Yes, again this one. Again you collected this thing on dead decaying trunk, right? I am sure that I am correct. And they are the species of Lentinus, Lentinus, right? It's a gilt fungi growing on dead decaying tree trunk, right? Then it is difficult. It is difficult. This is it is not mature. It will take completely different shape, and it is not possible. Achha. Yes, this is this is another species of again Coprinus or Coprinellus. Yes, it will. This species probably it will take a different shape after some time. The whole thing will be black, and it will produce black spores again. Where from you collected this species? From near locality?
Yes, no.
Nearby locality? Okay. Yes, this one growing on soil. Could you tell me this one grow? Where did it grow? On soil or tree trunk?
It is on soil.
It is on soil. Yes, it is. If it is on soil, then it is very difficult, because you know that I have to observe so many characters. Otherwise, it is not possible for me to say now. So yes, this one again Coprinus. You have collected lot of Coprinus species. See here already it is changed to black. This is another Coprinus. Yes, this one. Yes, if it is pink spore, pink gill. Yes, if the gill is pink, there are 3 to 4 genera, prominent 4 genera, which are found in tropical belt. One is Volvariella, highly edible species, growing on straw, and the second one, you know, Termitomyces, growing on termite beds, highly edible. Every in all the tropical belt we find Termitomyces, and then another one is Pluteus, another one is Macrocybe. They are pink gill. So here only a single fruit body is presented to me. So it requires more sample to identify the species. So না অক্সিডাইজড হওয়ার পরে পিঙ্ক হতে পারে। অক্সিডাইজড হওয়ার পরে যদি হয় তাহলে তোমাকে একটা জায়গায় টাচ করে দেখতে হবে যে সেটা শুধু অক্সিডাইজড হলো কিনা। এখানে তুমি দেখো যে যতগুলো গিল আছে সবাই পিঙ্কিশ। হ্যাঁ, সবাই পিঙ্কিশ মানে তাদের ক্যারেক্টারিস্টিক্স ফিচারই হচ্ছে পিঙ্কিশ গিল, পিঙ্ক গিল। ঠিক আছে, তো এটা একটা দক্ষযোগ্য অনেক অনেক স্পিসিস যেগুলো আমরা কিছুই এখনো জানি না, এবং আমাদের ইন্ডিয়ার যে ওয়েলথ সেটা সম্বন্ধে আমাদের কোনো আইডিয়া পর্যন্ত এখনো নেই। আমাদের যত মাইকোলজিস্ট যারা আমার আগে কাজ করে গেছেন, এবং তাদের এটা কোনো ভুল নয় বা তাদের কোনো দোষ না, কিন্তু আনফর্চুনেটলি what we have done, we have misapplied the American or the European name for our species, which are actually wrong. তো আমরা যেগুলো জানি এখন সেগুলো সব পাল্টাতে হবে। আমি আশা করি যে তোমাদের মতো স্টুডেন্টরা যারা ভবিষ্যতে সায়েন্টিস্ট হবে তারা নিশ্চয়ই আমাদেরকে নতুন দিক দেখাবে, এবং তোমরা অনেক অনেক স্পিসিস জানতে পারবে এবং লোককে জানাবে, যাতে আমরা মানে রিচড হব। থ্যাংক ইউ। ইটস ওভার।
Thank you, sir. We are really grateful to gain so much knowledge from you. Now, I would like to call our respected Dr. Onulina Manna ma'am on stage to present the gift to sir. Ma'am, please.
Thank you, sir. The today's first session is over now. Now we will take our lunch. So students are requested to take their lunch at the respective place. After completion of our lunch, we will start our second phase of today's journey. Thank you to all of you. That's yes.
Good afternoon to everyone. It is the second session of first day's workshop. This session will lead Dr. R.K. Gupta, scientist, identification of algae. Dear student, most of you know the Indian phycologist, father of Indian phycologist, M.O.P. Iyengar. The maximum contribution in this phycology came from the South India, eminent phycologist. And you know that from biofuel to charcoal, biochar, then food to nutraceutical, at the same time omega-3 fatty acid and other essential goods which are needed in our day-to-day life, we got from this algae. That's why generally in my class I told you that our future destination would be the algal kingdom, because carbon sequestration, at the same time oxygen generation, the major part played by these algae. I am thrilled now to introduce our respected scientist, a brief CV. Dr. R.K. Gupta, Scientist E, Head of Office, Central National Herbarium, Botanical Survey of India, Howrah. He joined BSI as a junior scientist, assistant at Shillong on January 1991. He got his PhD from the Ranchi University. His PhD topics was taxonomic studies of Euglenoophyceae and Dianophyceae of Ranchi. His specialization in algology. He published 5 books, 4 book chapters and 92 research papers in reputed national and international journals. He visited UK in 1996 for planning a project in biodiversity conservation, and in 1998 he visited Antarctica for studies on datum. Thank you, sir. We are eagerly waiting to learn something, and we highlighted the ourselves, knowing from some knowledge from your end. Thank you, sir.
Respected faculty members and my dear student, in this session we will discuss about the algae. Actually, algae are the very simple photosynthetic organism that lack embryogenesis. Algae are ubiquitous. That means it found in everywhere. Where light and moisture is there, algae will be grow there. And particularly for the beginners, algae you can divide microalgae and macroalgae. Macroalgae normally inhabiting in the ocean, and microalgae normally appeared in freshwater habitat. These are the number one. Number two, so if you go to the habitat wise characterization, it can also divided into 2 parts. That is a stagnant water or in free flowing water. For stagnant water, for example, is pond where water is fixed. It is called lentic type of habitat. Another one is the flowing water condition. Some algae are growing. So that particular type of algae is called exotic algae, lentic and lotic algae. And if you make a comparison, stagnant water, that means lentic water, algal diversity is more as for comparison to the running water. Likewise, if you calculate its diversity, so microalgae number is more, and macroalgae, mostly the member of Rhodophyceae, member of Phaeophyceae only exist in the sea condition, commonly called a red algae or in brown algae. Third one is some algae are planktonic in nature and some are benthic in nature. So in planktonic algae, mostly minute algae growing in the surface of the water, and which is very, very suitable to understand the climate change. Microalgae, planktonic algae, and another one is benthic algae, means growing in the depth of water. So these are the very simple technique to understand the algae. So it found in the freshwater soil and rock, and some algae are very easily growing in the surface of the bark of the tree. So bark algae is the very, very important habitat. So I have reported about 40 species on the tree bark, particularly of Trentrifolia and Cytonema. So bark algae is a very important aspect. So it maintains the internal temperature of the tree habitat, tree species, and protect from the mites and insects. So this is the very, very important aspect of study. So unfortunately in India, very, very little amount work has been carried out of the bark algae. See, in this slide, I want to highlight here those which are marked in the green. So these areas are fully explored. So algal flora of Ladakh, written by Sasikant and Pushpa Gupta from University of Jammu, and Jharkhand, in algal flora of Jharkhand, written by me, and those which are marked in a red, very, very sporadic work has been carried out very little, not a very consolidated work appeared. And the rest part of the country, very limited work has been carried out. And in this connection, since survey documentation is the main mandate, we are focusing on the various habitats also. So, algae are growing frequently in the thermal spring habitat also, where temperature is above 85-degree temperature. So, in this condition, mostly blue-green algae are growing there. In India, if you review the literature of the thermal springs of India, around 345 thermal springs are there. And out of this, mostly found in Uttarakhand region. While writing the algal flora of Jharkhand, I have encountered 4 thermal springs. So, actually, thermal springs are the very specialized habitat, characterized by high temperature of water and surface inhumations. In West Bengal also, in very near from, I think in this place, Bakeshwar, thermal springs are there. So, Vishwa Bharati Shanti Niketan, Professor Sumit Roy and his team contributed a very good paper on the Bakeshwar thermal springs. So, what happened? Thermal springs, actually main kund are there and its outflow is there. In main kund, mostly blue-green algae appeared, and its outflow, where temperature is little bit down, so another group of algae, members of euglenoids and some diatoms you will encounter. But in the main kund, mostly blue-green algae. One more thing, so you can also categorize algae, the prokaryotic forms as well as in the eukaryotic forms. Prokaryotic forms, that means its nucleus is incipient. Incipient means nucleus is not bounded. That's called prokaryotic algae. Parallelly, in prokaryotic algae, mostly blue-green algae, and other than blue-green algae, all algae comes under a eukaryotic. See, for the collection of phytoplankton, I have already spoke कि it is a minute algae growing in the surface of the water. So, simple method is you use a plankton net. First and foremost thing is to collection. So, different mesh size are there, 10 micrometer, 20 micrometer, 40 micrometer, or even then 5 micrometer. Just you hold these things and pass water. All algae will be trapped in your net. Another method, if you, during collection, if you use boat, then backside of your boat, just you fix your net and boat will be running, and within no time you will collect a number of microalgae. So this is a very simple technique. So at present moment in India, 9,098 species are reported and described, which are reflected under 206 families. And among them, green algae are the dominating algae. So, since the green algae are dominating, so I have used Frisch system of classification. It is a widely accepted throughout the world. So, green algae are divided into 9 order. So, volvocales to kerels. In the member of volvocales, flagella is the dominating character, and I think you are a BSc or MSc student, na? So volvox, Chlamydomonas, Carteria. So these are the common example belonging to the member of volvocales, and then member of volvocales, then member of chlorococales. In this group of algae, flagellal part is absent. Third thing is eulotrichales, means unbranched filaments. The common example is Eulothrix. Its chloroplast is a girdle-shaped chloroplast. If you put a single drop of iodine, it will purple bluish in color. So for identification of the green algae, so 4% iodine will be used, because this group of algae having a starch quickly react to iodine and turn into the purple brown. For clear cut visibility of its shape and the size of chloroplast. Fourth one is the member of cladophorales. Here filaments are there, but filaments are branching in nature. The common and typical example of cladophorales, cladophora. I think in your course, cladophora must be there. So, then member of Chaetophorales. Here also, in this group, a heterotracheous growth are there. Heterotracheous growth means the prostrate system followed by the erect system. The common example of member of Chaetophorales is the member of Trentrifolia. And in this group also, in Chaetophorales, one very peculiar algae that is called in parasitic algae, that is a Sipulurus, mostly preferred to grow in tea leaf. But Some reports are also there in mango leaf also. Cephalurus are growing. In Cephalurus also, prostrate system and as well as in the erect system. And it's commonly called red rust of tea. When the tea plantation is affected by Cephalurus, a dotted patch will be occurred, and the remedy, just you cut the twig and burn it. This is the only a solution. It is a very difficult to growth the Cephalurus in Thea sinensis, that is a tea leaf. Another is member of Odogonales. In member of Odogonales, a cap cell will be appeared. Only 3 genus is occurred throughout the world. That is a Odogonium, Odogonium bulbocate, and Odocladium. Odocladium grow in the moist surface. Then member of conjugales. So I think you have seen in your laboratory Spirogyra, Gygnema, Mojaceae. So these are the very, very common, Dibera, these are the common genus. And scalariform conjugation is appeared. Sometime lateral conjugation also occurred in this group. And parallelly to this, in conjugales, desmids are also classified under this group. So, desmids are also indicator species. Mostly it found in oligotrophic nature of water. Oligotrophic nature of water, that means very low level of nutrition are there. And water should be stagnant, then desmids will be growing there frequently. So this species or this group is also very, very useful. Nowadays scientists are using this group to make some data related to the climate change. Not a single species of desmids are reported in sea condition. Member of siphonales, a cynocytic mycelium like growth are there having no subtation, and the common example is Vaucheria. And advanced one is the Carels. In this green algae, node and internode systems are there, and it is considered that it is an advanced green algae. Cara, Nitella, Tolipella, Thamnohymnion. So these are the very common genus, and it is found in the small pool, puddles, ditches. I think anyone has collected Cara? बताओ कैसा होता है, आप बताइए. अरे बस 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 बैठो बैठो, सही बोल दिया आप. बस 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 आगे मत बताओ. एक्चुअली हमको बहुत अच्छा लगा, उसमें जो रिप्रोडक्टिव बॉडीज है ग्लोब्यूल नोक्यूल है, जो कि थोड़ा सा डीप ब्राउनिश कलर का है. एंड जैसे ही इफ यू ऑब्जर्व अंडर योर माइक्रोस्कोप, you will take measurements and its position, then species will be, and you fit your character to the key and easily will say this is a Kara, this species. You have collected from ditch. Which type of habitat? बहुत बढ़िया, बैठो, अच्छा लगा भाई. So I have already told about the prokaryotic and eukaryotic nature. This is very, very basic concept to stimulate the interest towards this important group of algae. So prokaryotic, Cyanophyceae, then you see in 1b, eukaryotic. Eukaryotic, in the total things are there. So actually we have written one paper, so definitely I will share this paper to all of you. So how to know the freshwater algae in India? This is a very important paper, and this paper was published, one book published in Calcutta University, Professor D. Mathi and his team. So this thing, this paper included. So definitely, sir, I would like to share this paper, and you make a multiple copy, and I will request to distribute to the students. So this is a very useful for academic point of view, as well as if you're facing interview, UPSC staff selection, so this paper will be very, very useful and written in a very simple language. So I will give this paper to our NV center, and definitely Tapas Saab will share to you, sir. Then if you go the life cycle of the algae, broadly asexual and sexual forms. So in asexual, everybody knows spore formation and fragmentation multiply algae like anything. And isogamy, heterogamy. So these are the life cycle, or you say the alternation of generation. So all algae are gametophytic in nature, then zygote, then meiosis occurred, and then germinate to the haploid plant. So 5 type of life cycle occurred. In algae. I am talking about the bark algae. Actually, you see, this is a tree of Samania saman. This photograph I have taken in our garden. The whole trunk, green mat-like structure are there. Okay. And if you tease a single filament and observe under microscope, I have already spoke, it is a heterotracheous in nature, prostrate system and erect system will appear. See, this is the microscopic structure of Trentrifolia. And if you see here, some brown patch is also there. Although it is a green algae, but due to excessive amount of hematocyanin, so some orange spot is also occurred due to its pigmentations. Yeah, another algae, planktonic algae, Microcystis. See, all these photographs related to bark algae, so I have taken from on our garden. And in garden, so banyan tree is there, and more than 5,000 prop roots are there. If you watch carefully its prop root, so you will find a green mat will be appeared. The green mat is nothing but it is a tentrifolia, and in banyan tree prop root, this species occurred throughout the year, I have noticed. In mahogany tree also, because in mahogany tree, so bark is different ridge shape and other, it provide a very suitable growth. So in this, another other than tentrifolia, cytonema is mostly growing in the tree of mahogany, trunk of mahogany. So I have given a brief thing about the thermal spring also. Actually, Kritikar is the first worker who published a paper in algae in 1886, first Indian worker, and his contribution appeared in Journal of Bombay Natural History Society. First mane Indian worker, his contribution appeared. And as far concerning the thermal springs in Assam, Bihar, Gujarat, Haryana, so Uttarakhand, so these are the places where thermal springs number are more, and if you count, about 345 thermal springs are reported, dominating by in Uttarakhand region. In Badrinath, Chamoli and all the track, you will find a number of thermal spring in a several distance. And in Maharashtra, so in Maharashtra also, very important contribution has been made by Ila Gonjalvis and his student. See, in thermal spring, all are blue-green algae, and in this algae, very thick mats are there. So, there is a possibility of very high scope to work in thermal spring. If you explore in your nearby in Bengal or in nearby in Odisha, so you can plan accordingly. So these are the mats. So in Jharkhand, one district of Jamtara, so because in Jharkhand total 24 districts are there, and I have covered all districts, and 744 species are documented. Well, and this well water is boiling like anything. These are all habitats of thermal springs. One interesting thing in these mats, if you 5 centimeter or 3 centimeter mats are there, if you observe the upper surface, species composition is different. In middle part, species composition is different. In lower part is entirely different. So it grow as a tire in a series form. So these are the some micro habitat, Fragillaria, a diatoms. For diatoms, a navicula. So nowadays, diatoms play a very, very significant role. And its cell wall is made up of silica. For identification of diatoms is not a so easy thing. So you have to have clean its diatoms frustule. Actually, diatoms di means two. Two cells are fitted together. You understand? Two cells are fitted together and full of silica and other calcareous material. So for identification, if you want to identify the direct collection, so your identification will be wrong. So for diatoms study, it need to be thorough clean by acetic acid, and you just boil for a while, and then you centrifuge your things. A pellet will be observed. Just you decant the things and see under microscope, you will get a number of diatoms. So in diatoms biology, in India, Professor H.P. Gandhi and Desikachari group of Madras made a very, very valuable contributions. Desikachari Saab is the student of Professor M.O.P. Anger. Actually, if you look, because I have visited several times to University of Madras in Guindy campus and see the Professor Anger lab and Desikachari lab. So still these legacy were maintained by their faculty member. Actually in 1932 within India, Anger and Professor Bhardwaja, B. Bhardwaja went to England and training he got under Professor F.E. Fritsch, and after returning to India, they have established a very strong school of algae. Anger established a school algae in Madras, and Bhardwaja established a very strong school of algae in BHU, Banaras Hindu University, and their students established a small laboratory in the different places. So these are the photomicrograph of blue-green algae. And you set up a very small study having no expenditure, but your result will be very fine. Several times I have spoke about the thermal spring. You categorize the water on the basis of your thermometer. Then you will find in between 45 to 50 degree, the thallus texture is little bit soft. But when temperature is high, it is a leathery form, and in the above 80 degree, it is a leathery form of things. So, on the basis of temperature, you observe your element. So, these are the very simple experiment. You can test this thing if you are interested to the thermal springs forms. Then parallelly water analysis is also a very important factor. Basic data like pH, electrical conductivity, total dissolved solid, DO. So these are the common things. So you collect algae parallelly. You observe these things also. Nowadays, water analysis kits are available and very in student rate, in very cheap rate. So for beginners, so I will suggest कि if you are really interested to study algae, parallelly you record its water quality also. So this thing will be help when you go for publication. So some ecological notes is very important. Only identification of species, okay, these are the baseline, but you give some momentum. You put some additional data to make your paper user friendly. So some algae forms bloom also. Bloom means, have you seen in your field visit such type of blooms? Actually, blooms are a single species growth in excessive forms. In this time, it doesn't allow to grow another plant, another algae. This is a slide of Microcystis aeruginosa, although it is a poisonous algae. So, when it grows, the DO level, dissolved oxygen level will be go down drastically, and death of fishes occur frequently. So these are the small parameters to understand the climate, and you correlate your work. I am not talking about the very big data. Your small effort will throw some light. This is another bloom of Euglena sanguinea. You see, all these patches of a single species. And very interesting thing, I will share about this photograph. If you take photographs in daytime, in a very scorching sun, its color is like a brick color. But you visit same habitat in the evening, water color is normal. So, in this condition also, in this species Euglena sanguinea, hematocyanin pigments are very, very high. So, I have already discussed about the macroalgae and micro. So, previous one is the micro, and these are the macroalgae. Caulerpa, Pedina, Joneria, Alva. So, although this photograph is taken by in our herbarium sheet and growing in the sea condition. Comes under macro. So, cleaning of diatoms is very, very essential. So, with the help of acid wash, it can be possible. So, these are the all these photographs taken by in our SEM. So, broadly, diatoms are divided into two parts: centric diatoms and pinnate diatoms. Centric diatoms means, broadly to understand, a rounded shape. Comes under centric. When structure is slightly bit oval, that comes under pinnate diatoms. So all these photographs has been taken under SEM. This example of centric diatoms. Just you see the structure. Then Gomphoneema, different shapes are there. And for identification, these are the ridges. You count for identification, length and breadth is the very common thing, and this structure is called a styrene. You also count the styrene, how much it is, for correct authentication. All these photographs under SEM in electron microscope. Another group is the dinoflagellates. So, in this thecal plates are there. On the basis of its plate counting, species will be authenticated. Gymnodinium is the common example. Peridinium. Yes, Serasium is a very important alga. So, these algae, particularly this Serasium, is very common in Ganga water. Very, very common. Just you tow your plankton net and take out these algae in your petri dish, and directly you see under your microscope. So seresium, this type of a structure, horn shape, you will find. So आपको हम ये बताया था ना वो कारा, ये देखो, ये देख रहे हो आप, its reproductive bodies is a very advanced green algae having a clear cut nodal and internode systems are there. So these are the some photographs of member of euglenoids. Its reserve food material is paramylon. Member of Xanthophyceae, member of Chrysophytes. So dino brown, these are the very common algae. Cyanura, Melomonas, and these are the some important desmids, and I have already spoke, it is grow in the oligotrophic water, having a low nutrient value. And these are the very ecologically important group, desmids as a whole. Some filaments desmids, although these filaments desmids are very, very rare in nature. But although it is growing in Indian water. The common example is Desmidium, Halotheca. Mostly desmids are a unicellular, but some very few desmids are in filamentous in nature. So according to Fritsch, algae are divided into 11 classes, Cyanophyceae to Rhodophyceae. Some important economic importance also, and used in a medicine also. Medicine means spirulina tablets frequently available in a chemist shop, particularly those who are a vegetarian. So spirulina tablet is very, very useful. And algae in the role of agriculture. This create a job opportunity also. Job opportunity means in IARI, New Delhi, they are preparing biofertilizer with the help of heterocyst algae, heterocyst blue-green algae. What are those? Enavina, nostoc. Actually, this laboratory was established in Delhi in Pusa by G.S. Venkataraman, and in 1959. Since then, this legacy was maintained, and they are preparing a biofertilizer, and annually they recruit a scholar either in a project or in a regular job. So you can check its website also, IARI, Indian Agriculture Research Institute. So this is the very simple group of plant. They can change your life. Change your life means कि a job opportunity scopes is also occurred through this plant. This is the number one aspect. Number two aspect, they are making a very huge collection of algae in pure forms. So, in this connection also, in culture collection and in a biofertilizer, these are the two major wing. They recruit a person in a bulk level. Either in a form of a scholar or in the form of as a regular employee. So several times I have visited there and I have seen, and they prepare biofertilizer and distributed to the farmers free of cost. Time to time they arrange Krishi Mela in the different localities. So very huge program is running. So that's why I have experienced कि with the help of algae, with the study of algae, it creates a job opportunity also. Now this thing is very important for the youngster also. Then bioremediation, algae as a food, some macro algae. If you visit a seashore, particularly alva, enteromorpha, you direct collect from there and just wash it and cut into the small pieces, and you take as a salad also. And even in the coastal belt, particularly in Gujarat and Maharashtra, algal soups are very common nowadays. Alva soup, pedina soup, juneria soup. So I have seen and I have taken also. So what is this? This is a creating a small opportunity, and this is very needed. And after taking immediate you will feel very energetic. And I have seen also in Gulf of Mannar in Tamil Nadu. So in industry also, in particularly in cosmetic industries, algae used like anything for making shampoo, soap and other things. So these are the some common algae belonging to the green algae, Gygnema, Maugesia, Udogonium. So which type of genus is included in your course? Tell any person. Tell 1, 2 genus. I will focus on this also because it is helpful for your examination point of view also. Very fine. Bus, yes. Sometime in 5 marks or 7 marks notes, examiner will ask Polysiphonia. Write a brief note, 1 or 2 lines. Definitely Polysiphonia is a red algae growing in the sea water. Okay, then pit connections are there. So these actually examiner expected to student should write something in the bullet points. Kara, I have already told, and you're one of the students, I am very happy. Globule, nocule, node, internode, and it is a green algae. Then, member of Udogonales, I have told presence of capsule. These are the rigid character. So, these are the points. So, for examination point of view, followed by, I have thrown some light on the job opportunity also. Actually, I would like one more thing in between this about the history of algae. Actually, what happened in India, if you go through actually one IS officer, his name is MS Randhawa, Mohinder Singh Randhawa. Earlier days, it's called ICS officer. Now it's called IAS officer. So in present context, basically he is a IAS officer, but he has a very, very keen interest towards algae on those days. And he make a plan to write the monographs of algae. And that time Randhawa was in Agriculture Planning Commission Director in very top post. That time he catch MOP anger and assigned some task. Then not only anger, HP Gandhi, Gonjalvis, that time top algalogist he contact. So I will provide all support to you about the fund, about the facility. Because that time Randhawa was in very top position and he is very deeply interested in algae. And that time he hire around 10 to 15 top algologists of India and provide all facility, fund and other things. And those peoples are write a monograph. Nowadays, all these monographs are frequently available in IARI cell counter library. And still its price is 10 rupees, 2 rupees, 5 rupees. Government of India is not revised. And if you expend 100 rupees, you will purchase large amount of books on algae. And still available in IARI library. Written by Anger, Desikachari, Ila Gonjalvis, M.R. Saxena, H.P. Gandhi. So, and that time also, chunki Randhawa was basically an administrator. So, during daytime, he is very busy with his administrative work. But when he free from his administrative work, particularly in Sunday, he identify all these algae and during his official tour, he collected several algae from the Faizabad district and other places, Almora, and night time he observed under microscope, and finally he published Xanthophysi in India in 1959. Then Anger group contributed, Gonzalez group contributed, like this, like this, like this. So for the progress or development of the algae, so MS Randhawa play a very, very key role. So this is it suddenly came in my mind, so that's why I have shared these things. So for identification of algae, nowadays no doubt several hi-fi microscope is available. We are using in BSI also in a Nikon microscope with microphotographs, scales and other things. But for taking a line diagram is the very old technique. So we are still using, apart from the micro photograph and parallelly to the line diagram also. Sometimes algae are overlapping, so its photography, if you take, it appeared a blur, not clear. So that time we are using a line diagrams also. Member of Euglena. This is all the member of Trachlomonas. ये iron indicator plants, Trachlomonas. Its identification view, these are the collars, sometime collar erect one and sometime the depth. So on the basis of this type of things, by practicing, you identify your material. I have already spoke about the paramylon, a reserve food material. So microalgae we preserved in our CNH in a liquid form. 3% of glycerin, 3% of formalin having 1 drop of glycerin. 1 drop of glycerin is very, very important. It's minimize the evaporation, because in bottle forms, if you preserved algae without glycerin, over a long period of time, it will be dry. And in this condition, if is algae or dry, it cannot be rehydrated. So what we did, so 3% formalin having 1 drop or 2 drop of glycerin, minimize the evaporation. So you can save your material for a long time. This is a very simple technique for long-term preservation. And you 1 or 2 pinch of, you also add copper sulfate for its color retain. I think you have collected algae in the field and over a period it turns white and yellow. So I think you have not included the copper sulfate. You 1 or 2 pinch copper sulfate, then your color will also intact. So these are the some centers of algae. So you see, these are the books. Actually, in the leader of Dr. Randhawa was appeared volvocales chlorococcales, eulotrichales, udogonales, gignemales, carels, vaucheriales, and cyanophyta and Dinophysi of Indian Sea. These two books are written by Desikachari, and all these books are written by Iyengar, Desikachari. This one is M.T. Philippos, Gonjalves, Jignemesi Randhawa himself. So these books are particularly 1 to 7. I have told कि less than 100 rupees you will collect all these books. Simply you go there and ask, he's in cell counter division. So to stimulate interest towards this group, you will collect a large number of literature, almost in free of cost, almost. And BSI has already also taken a program to write the Algae of India. So in this connection, we have published 4 volumes of Algae of India. Actually, this program to write the Algae of India, this program was designed by our former director, Dr. Sanjappa sir, and under his leadership, this program was finalized to write the Algae of India, and we have till date published 4 volumes. Apart from this, so flora work is also published, and all these books are also available in our cell counter here also. In CNH, we have established a cell counter for easy access to the researcher and beginners. So all books are available not only in algae but in the protected area and the flora book, identification manual, several kind of books we have sell here also. It is I think 3 year back. Earlier our cell counter earlier in Salt Lake in a headquarter office. So I think 2 or 3 years back we have established here also. So these are the something about the literature. You see, it's some photo micrograph taken by under a Nikon microscope. These are the Odogonium, Spirogyra, and these are the Navicula, Rhodophyton, diatoms. So this all about in this moment. So student, ask your any doubt or any question, it is welcome. And I will tell one thing, BSI is always ready. This is our main mandate also for authentication. So don't feel hitch. You contact BSI. So definitely our scientists will provide a full support, particularly the scholars and faculty members.
Sir, many thanks for your valuable information. Particularly, I am saying to you that India is a highly populated country. Unemployment is the burning issue in this moment.
Yes, that's why I have thrown, sir, some light. So these websites are easily available in your Google. So it is in twofold. For making biofertilizer as well as in its algal strain center, collection center. So in these two divisions, they recruit a people.
In this regards, I would like to address this problem through you. Why not Government of India are not taking this initiative step, particularly procurement, packaging, exporting, and in this scenario, how much amount of foreign money we are earning by exporting?
No, definitely, sir. Government of India is always initiated, and nowadays for the employment also in every sector. Now the process is slow, no doubt, but it is not a stop. For making spirulina tablet, so actually these spirulina tablets are small pharmaceutical industries. So in industry, not a single man will prepare the whole tablet. Definitely whole teams will be there. Some are collecting the material, some are purifying the material, some are processing the material, some are packaging the material. Then it will come into the flow to the market. So my thing is, so process is initiated, sir, and all the question is to give a strong momentum. So time to time we are also addressing these things, and
just few years ago, I go through this English paper. A foreign phycologist visited our country, and he mentioned that India is a vast water bodies, and there is a huge scope.
Yes, I fully agree, sir.
So this algae cultivation, that means algae domestication, like crop domestication, it is needed. So,
sir, I have told you, sir, in just a Gulf of Mannar, so algae plantation is also occurred, sir, through rope method. Because for making a soup, for making a salad, they want large amount of algae. So if the technique is very, very simple. Very simple, sir, plantation in the sea. Just suppose one rope is there, sir. Rope. You turn anti-clockwise. Then gap will occur. Suppose you are interested for alwa. You put alwa, and in water, just you tag this rope and down into the sea water. Within 20 days it will grow, and over a period of time this rope technique was slightly expensive. So what they did, they used a hollow net, hollow plastic net, sir, and they put algae there, and these nets they kept in the sea water, within a month they will full your net is full of algae. So if you go through the critically the videography or video, so mass algae cultivations and for preparation of these things, so then you will think something is going on towards algae for the betterment of the life.
Sir,
For mass cultivation, I am talking these things.
another burning issue is that plastic. Why not we are preparing this bioplastic from the biomass of algae? There is a huge scope. Already it has been started in.
Definitely, sir, scope are there. Nowadays, we are also in a mission mode to reduce to less plastic, single-use plastic. But it takes time, sir.
So,
Government taking all steps, sir, regarding this.
sir, you are in the big platform and we are at the grassroots level. Just I am saying this suggestion to the government of India.
Person is same, sir.
Government of India, please convey this message. We can overcome this issue, that is the use of plastic.
You see, sir, I will giving a very small example. In our botanic garden, it is a 273 acre. If you even then you take this bottle and if you enter in our garden, it is not possible. Immediate our security will catch you. Although you have a valid tickets and all things are there. So we are taking all possible measures, sir, to reduce the plastics under BSI, and nowadays Mission Life program is also a part of the program of government, and we are following all these things. So definitely mind will change, sir, and time will come and it will solve, sir.
Sir, one interesting point is that from my curiosity level, I read that in European country where the pollution is alarming in stage, like street lamp, they are making aquarium. As the algae sequestration of carbon dioxide is higher than the land pan, that is why they are using as a street light, that is forming the aquarium. Why not in Delhi? Most of the time that we see or watch in the television that Delhi is a highly polluted country. The
No
school
doubt, sir. Delhi, to everybody knows, sir. In almost Delhi water quality index, and Delhi is the topmost. But things are going on, sir, but in a slow method, and it need to be addressed in a big momentum form.
Thank you, sir.
Thank you.
We are the aspirant. Dear students, if you have any question, please ask our resource person. Any question?
Acha, okay, student, if you don't have question in present moment, so सोमेंदा ये प्रेजेंटेशन तो उनको दे देना है, और कुछ भी दिक्कत होने से आप एनवीज में आप कांटेक्ट करिए. सो डेफिनेटली वी विल प्रोवाइड ऑल सपोर्ट. I have already told, particularly the student, I always with him. Thank you, sir.
Sir, for your kind information, very recently I collected a algae from Kongshabati river, that is the Coel estrella. Coel estrella. On this algae, I got my PhD from JIS University. That is why I am interested in algae.
Acha.
Thank you, sir.
Thank you, sir. No, no. Thank you, sir. जरा देखो तो इसको.
Thank you, sir. It's been a great privilege. It's been a great privilege to take so much knowledge from sir. And now I would like to invite our Honorable KD sir on stage to present the gift to sir. As we have headed towards the end of the program, I would like to request our Honorable Coordinator Sir to come on the stage and to give some Thanksgiving word to us. Sir, please.
গুড আফটারনুন টু অল অফ ইউ। অনুষ্ঠানের প্রায় শেষ পর্যায়ে আমরা এসে পৌঁছেছি। তো হচ্ছে আমরা এখানে অনেক এমিনেন্ট সায়েন্টিস্টদের অনেক কিছু বক্তব্য আমরা শুনলাম, যেটা হচ্ছে আমাদের যারা ইউজি এবং পিজির যে সমস্ত স্টুডেন্ট তোমরা রয়েছো, তো তারা আশা করি সেগুলো অনেকটাই অনুধাবন করেছো এবং বুঝেছো। এগুলো সম্পর্কে আমরা যে সমস্ত মেটেরিয়ালগুলো আমরা পাবো সেগুলো হচ্ছে তোমাদের যে হোয়াটসঅ্যাপ গ্রুপ রয়েছে সেখানে আমরা প্রোভাইড করে দেবো। ফার্স্ট অফ অল আমি ধন্যবাদ জানাতে চাই আমাদের ডক্টর সৌমেন গাঁতাইত স্যার এবং হচ্ছে আমাদের জালাল স্যারকে, যারা হচ্ছে মেন এই প্রোগ্রামটার হচ্ছে মেন উদ্যোক্তা। এবং মানে বেশি করে ধন্যবাদ জানাতে চাই জালাল স্যারকে, উনি এটা আমাদের সঙ্গে পুরো এই প্রোগ্রামটাকে কোঅর্ডিনেট করার জন্য অনেক রকমভাবে হেল্প করেছেন। এবং আমাদের সমস্ত ফ্যাকাল্টি মেম্বার যারা আজকের এই প্রোগ্রামটায় উপস্থিত থেকে শেষ পর্যন্ত চালিয়ে গেছেন খুব সুন্দরভাবে, এবং বিভিন্ন কলেজ থেকে আগত, ইউনিভার্সিটি থেকে আগত আমাদের স্কলার, টিচার এবং হচ্ছে ইউজি এবং পিজির সমস্ত স্টুডেন্টদেরকে এইগুলো খুব সুন্দরভাবে, শান্তভাবে, ডিসিপ্লিনভাবে শোনার জন্য অনেক অনেক ধন্যবাদ। এবং কালকে আমাদের এই প্রোগ্রামটা শুরু হচ্ছে ঠিক একদম সকাল 10টায়। সকলে হচ্ছে এই সময়ে এসে উপস্থিত হবে। থ্যাংক ইউ।